Hello, everyone, and welcome back to Creation Myths. Today, I have a really fun conversation to show you. This is actually a rebroadcast of a live show I did with Dr. Chris Thompson on his channel on January 9th, 2025. So his channel is linked down below. If you are subscribed to me and like what I do, you're going to like what he does. So make sure you go down right now. You could pause the video and then go down, click that link, subscribe, then come back and finish the video. That's Dr. Chris Thompson. So I chatted with him on January 9th. It was a really fun conversation. We talked about molecular clocks and phylogenetics and common ancestry, and it was also a bit of a prep session for a debate that Dr. Thompson did with Dr. Kent Hovind on January 11th, 2025, and I'll also link that down below. So this conversation was really fun. I enjoyed it. I hope Dr. Chris Thompson and I get to talk again because I would love to continue this conversation. And there was actually stuff that we talking about that we didn't actually get to. So I look forward to future kind of, continuing this conversation down the road. I hope everybody enjoys. Thanks for watching.
All right, we are live. So thanks everyone for being here today. And I want to thank Dr. Dan for being here from Creation Myths. So if you have not, I'm sure if you're watching this, you probably have seen his videos. If you haven't, you should. Dr. Dan is a professor at Rutgers University. He's an excellent science communicator. He has this YouTube channel called Creation Myths where he debunks and various aspects of creationism, focusing mostly on young Earth creationism, and also does debates. And he's a wonderful guy, does fantastic work. So I invited him on today to talk about a paper that I thought would be just an interesting way to spur a conversation. This is one paper of many that I've been reading over the last week or so in preparation for a debate that I'm having with Kent Hovind on the evolution of the carnivores on standing for truth. Okay, all of that sounds so goofy to just say. But anyway, thank you so much for being here, Dr. Dan. It was super appreciated.
Thank you. I'm happy to be here. And I think we do just have to appreciate the absurdity of two professional biologists sitting here having this conversation in the context you just laid out. This is...
Yes. In fact, that's what I plan on starting with, is that, for people that are just seeing this and who don't necessarily know me, I'm Chris Thompson. I'm a professor of neuroscience at Virginia Tech. And this is I'm interested in evolution. I have an undergraduate degree in evolutionary biology and ethology. For those who don't know, ethology is animal behavior. I started that degree at the University of Illinois because I was more interested in the evolution side of things. But I was also required to take some ethology classes, which then got me interested in mechanisms of animal behavior, which brought me into neuroscience. And now I'm a neuroscientist. So I'm not necessarily a trained evolutionary biologist. I don't work on evolutionary biology. And in fact, most evolutionary biologists consider debating creationists to be a waste of time. And they're not necessarily wrong, right?
Right.
But I think it's an important thing to do for some of us who enjoy it and are willing to do it. It's certainly not something that I expect evolutionary biologists to go out and do on their own if they don't want to, because it is largely a waste of time. And that is actually how I plan on starting my debate.
Because this is silly, and outside of this room where it occurs, nobody really cares, right?
Exactly.
Yeah.
Yes. Outside of this room where it occurs, and for the few random weirdo legislators that will be at the state house in some place that will try to pass some, say, like an intelligent design bill or young Earth creationism bill. These are things that could potentially be coming up over the next 4 years.
Or, the Speaker of the House of Representatives.
Yes, exactly.
Who has an author page at Answers in Genesis, if anyone cares to look it up.
I didn't know that.
Oh, yeah.
Oh, so he's contributed pieces to Answers in Genesis.
Oh, yeah.
That's not surprising given his, what is that program that he uses with his son?
The porn surveillance program.
Exactly.
Yeah.
Okay. Wow. Yeah, so he's in a totally different world, but part of that world, right? And it's the thing is, like, young Earth creationism while it's kind of a fringe movement, of course, it's still obviously something that you know, a lot of people believe in the United States and also in other places in the world. It's largely like the United States is the main place where you're going to have the highest percentage. Other than I think some of the surveys that include places like Turkey, it tends to be kind of high in Turkey because
Mhm.
obviously, like, there's a number of fundamentalist Muslims there as well. But their percentage is about the same as the United States. And then you look at Europe and Asia, like the percentage of people who believe in these things drops off quite considerably. So, yeah, it's a fringe belief, but it's big here in the US. So that's why we spend time doing it. And it's kind of fun. Like I like doing it because I end up learning an awful lot. And that's one of the reasons why, I reached out to you because I do know a fair amount of like how molecular clocks work, but that's not what I work on. I depend upon those things when I read them in papers and I just kind of take it as like these people obviously know what they're doing. I don't necessarily know all of the methods by which they're doing this, but because I was preparing for my debate, I wanted to learn more about it. So I feel like I know more about it now than I did, say, a week or two ago. And that even then, that still made me much more knowledgeable about it than like the vast majority of young Earth creationists.
Yeah.
So, and now I feel much more prepared, but there's still some things that I don't really know. So I appreciate you being here. And
Well, I'm happy to be here, and I also want to just add two things to that excellent
yes.
intro. One is that there's this kind of long running debate among real scientists of like to what degree should we engage with creationists, debate creationists, like share a stage with creationists, and for a long time, I think certainly I thought, and I think the consensus was that don't do that, don't like legitimize them
Yeah.
by giving them a platform was kind of the way people thought. I've done a complete 180 on this because the sad truth is they have a platform and they have influence. So either they're going to get to say their stuff uncontested, or there's a real scientist in the room that's able to push back.
Right.
I would rather the second thing be the case. And if that means I am that real scientist from time to time, then
Mhm.
okay. I think that is worth doing.
I totally agree with that, and I also have had that evolution, so to speak, of my perspective on it as well. I do think that it's important to have at least someone willing to do it. I agree that platforming, say, bringing in a young earth creationist to some major forum to have them discuss something where they're put on a prestigious platform, yeah, that's not deserved. But
Mhm.
if you go into their spaces, like I'm planning on Saturday, and arguing with them there and also giving them the level of respect that they deserve, right? Like, I'm going to be respectful, but not too respectful because we're talking about Kent Hovind after all. So,
Right. So
right.
that
Yeah.
level is
Yeah. I mean, he's not going to give
lower.
me much respect. So,
No.
like, I'm going to give it to him in return. And yeah. So, like, but they're doing it anyway. And they do have this influence. Like you
Yeah.
said, like the House of Representatives has a Speaker of the House who is listed on the author page of Answers in Genesis. Like
Yeah.
he is third in line to be president of the United States
Yep.
should the president and the vice president be killed in some sort of accident or something. He would be then president, I think.
Mhm.
So it's like, okay, yeah, like obviously this is a position held by people who have positions of power, and it's important to push back on it. And if we don't do it, no one else will. And I get why most practicing biologists don't think that it's worth their time because they're not wrong. Like there's a lot of other things that we would rather be working on than this. And if you enjoy, and also debating is not a skill that is
Right.
taught to scientists, right?
Yeah.
We do hash things out, but mostly it's done in a nice, polite way, discussed over papers or letters to the editor or at meetings. Sometimes it's not always so nice. I've seen some meetings where it gets really out of hand, but mostly it's done politely.
At least
But
with a facade of
yeah,
cordiality to it. Yeah.
exactly, sure. And so I don't know. I think it's worth it. And so like organizations like the National Center for Science Education, I'm a big supporter of them. I think that it's important to have someone pushing back,
Yeah.
and because otherwise they just get free reign. And the polling shows that it's what is it something like about 40% of the population believes in young Earth creationism in the United States, at least
And that's
the
and
test.
that's as
Yeah,
low as it's ever been, and it's
yeah,
still a plurality, I
right.
think. So
Yeah,
it's a lot. Yeah, and that
exactly.
and that's an important point that it is that large a fraction because it's not a standalone thing. We treat this, and I think the mistake a lot of I'll say a lot of us make when we enter this conversation is we're treating it like a scientific debate, which it is not. This
Right.
is the correct way, I think, to think about creationism and intelligent design, the intelligent design movement and everything,
Mhm.
is that it is a first and foremost a political project that is an arm of the broader right-wing Christian
That's
nationalist
right.
project, right? It
Exactly.
is part of that thing first and foremost. And the science language that the professional creationists like to add on top of it, that is window dressing. And
Mhm.
you look at people like Ken Ham, who runs AIG, he makes all these videos, he does all these interviews with, their in-house scientists and everything. Like, at the end of the day, he doesn't care. That's not what it's about. It's not
Right.
about the science. The science is just a piece of this broader cultural project.
Yeah.
And it's important to push back on that. And, so pushing back on the science is part of that.
Yes, I agree. The other thing about it is that, um, you know, the we if we give them the space to just have this sort of forum, where they can kind of create these institutes and just kind of do all this stuff without any sort of pushback or even recognition that this is happening out in circles outside of those, right wing media sphere circles. Um, then there's it just gets done in darkness, and there's no sunlight placed on the absurd things that they actually say, and then
Mhm.
allows like these people to sort of foster this alternative worldview, which is totally incompatible with what we know about how the world actually works.
Yeah.
And then it provides justification. Obviously, if you go all the way back to the Discovery Institute's wedge document, where they're going to use intelligent design as a wedge to break apart the naturalistic model
Yep.
of viewing the world. Yeah, I mean that is the goal actually of all of these positions of these people. I also think that one thing that we fail to do if we don't push back is one, allowing that space to be there, but also that there's a lot of intramural division within the right wing and creationism, especially within creationism,
Oh, yeah.
that we can exploit, right?
We
Like,
sure can.
right. Like, I don't think of flat earthism as something separate from creationism. Like the large justification for flat earthism is basically biblical based, which is exactly what young earth creationists do as well. Now, most young earth creationists sort of recognized how absurd it is to think that the earth is flat. Like, even I've heard Kent Hovind talk about like how goofy they are, but like, sorry, man, that's part of your team. And
Yeah.
kind of saying that, like, yeah, I think it's important for people to just acknowledge that part as well. That, like, there's a lot of interscene divisions that you have to be knowledgeable about, like say you and I are, that allow you to exploit those divisions. And that's obviously tough for practicing biologists to do because like they don't want to waste the time on learning about this stuff.
Right. It's a whole other
But
thing
it
you got
is
to learn.
undermining then the scientific process, and if we have kids that are not getting good science education in public schools, or if they're going to be, say, removing funding for doing the actual science or for teaching at the university level, like that's obviously going to have a huge impact on the scientific enterprise
Or
long term.
Or if, for example, just hypothetically, not that this would ever happen, but just hypothetically, if there's a once in a century pandemic and we rapidly develop a new type of vaccine, and you've got a huge swath of the population that is skeptical of science in general, that doesn't
Right.
want to take the vaccine because reasons.
Yeah. Right.
Hypothetically,
Exactly.
not that that would ever happen,
Hypothetically,
but
yes, or hypothetically that somehow horse paste is considered
yeah, right,
um, a viable method for dealing with
yeah.
viral infections.
Stuff has real consequences, folks.
Yes, it does. Yes, and so scientific literacy is really, really important. So, anyway, I really appreciate it. Totally on board with all of those things, and yeah, kind of gives me the a sense of kind of the larger context that I plan on doing for my debate with Hovind. It'll be my first time debating him. And hopefully my last. Honestly, I think everyone wants to take a turn, but then they realize like how pointless it is in some ways.
Just
Um, yeah, just do
that
it
guy.
and get it done. But
That guy.
but the reason why I picked the very sort of specific niche that I picked is because I wanted to force him to kind of get away from some of his talking points.
Yep.
He'll often talk about like, well, do you believe that you're related to a mosquito, or do you think that you descended from an amoeba, which I always love, like, because that just shows his utter ignorance of
You don't know.
exactly how evolution works.
Yeah.
No, amoebas
You don't
exist
know.
today. I don't think amoebas existed 4 billion years ago, right? Like, you really don't understand this. Yes. So, but putting it constrained within a specific group of mammals, the carnivores, gives me a lot of material to work with to illustrate some of the things of why younger creationism just simply doesn't work with the data that we have. And we don't have to go farther beyond even that. Like, so basically, I plan on putting this as a context that, like, okay, where the carnivores came from, I have my opinions and what my beliefs based on data that there are descendants of the carnivores and they evolved descendants of the carnivores, and the carnivores evolved from those descendants, and we can talk about that a little bit. But you could also just the whole thing could just be that they were specially created
Yeah.
64 million years ago,
Boom.
and yes, and now we're going to talk about descent and modification from then. Like, that would still be consistent, and so we're kind of holding it all to this one thing. And the reason why I pick carnivores is because they have all these different what they call kinds. We'll get to that at the kind of end.
We'll
So
get to
I've got
it.
yeah. So part of this, though, is talking about molecular dating methods. And so I put together a slideshow based on the paper that's linked in the description of this video. So you can go and click on that. It's a Nature paper. You should be able to find it in other places too. So this is a paper that came out just 2 years ago. And I got to present. Hold on a moment while I do this. Come back to SteamYard. I got all this set up beforehand, so I apologize. Thanks for everyone who's watching. Appreciate it. Be sure to hit like and the video. Yes, and obviously subscribe to Dr. Dan's channel too. So, all right.
The dance of windows on your screen.
Yes,
I don't know how many monitors you have, but I feel like I always need another one.
I've got 2. Yes, but it doesn't feel like enough. All right. So this is what I decided to start off with.
Nice.
Yeah. So that's Insane Clown Posse, true poets of our modern era. Famously were very confused on how magnets work. And I'm confused, obviously, on how molecular clocks work. So that's how we're starting off. All right. So this is something that I took off of the Berkeley website. They have a nice, sort of very helpful, really
Excellent
easy
website.
to understand explainer on aspects of evolution. And so this is just like real basic way of understanding how molecular clocks work, the idea behind it, the principle behind it. So if you have like some population, what they're saying here, around over 50 million years ago, there would be, obviously they would have a genome, and that genome would have a series of A's, C's, T's, and G's, the basic nucleotides that make up the genome. And stretches are, though, what we know from over time is that they're going to accumulate mutations over time, right? There are, actually from a lot that I've learned from you about neutral evolution, mechanisms of neutral evolution. And Kimura, was one of the first people to really define aspects of neutral evolution, but that this inevitably means that there's going to be mutations that do accumulate over time. Now, this isn't necessarily consistent across all parts of the genome, as your video from last night nicely illustrated. So I encourage you to look at that. But there's still going to be, it's a probabilistic thing where mutations happen. And then if they get incorporated into the genome and then passed down through generations, that's referred to as substitution. That's substitution rate. And correct me in any place where I'm wrong, because you're more the expert on this.
Yeah. So just with like mutations versus substitutions, the really simple distinction that everyone should know is when a mutation is just any change that happens, right? And you've got some in this illustration here, a T changes to a G, or a G changes to a T, that's a mutation, right?
Yep.
When you have a population and that mutation becomes fixed in that population, so its frequency is 1. So for that locus, that location in the genome, every instance of that locus has that mutation. When its frequency is 1, now it's a substitution. It's fixed in
Yep.
that population. And when we talk about things like molecular clocks and the time to the most recent common ancestor between different lineages, we look at the substitutions because those are the mutations that accumulate. So kind of a simple way to think about the mutation versus substitution rate is mutation rate is the rate at which mutations occur. Substitution rate is the rate at which they accumulate
Right.
within this or that lineage.
That's right. Exactly. Thank you for explaining that. I couldn't say it any better than that. That's exactly what this is illustrating. So over time, if we could take snapshots of ancient genomes, which we will get to briefly in a little bit, but if we could take snapshots, you can see that, like, okay, there would be these mutations that have accumulated over time, and that those are substitutions. But then as you go on through time, there's going to be more and more mutations that have accumulated that are differences now between these two very separate populations, probably two different species in this case, if we're talking 50 million years apart. Certainly that's the case. And that there are going to be these differences that have occurred that are apparent within the genome that are completely fixed. Those are substitutions. Then you have to find out what the substitution rate is, right? So the idea that there's this mutation rate that occurs at some sort of relatively consistent rate. That's known as genetic equidistance. It's the kind of that phenomenon of that there's going to be mutations that will accumulate in two separate populations over time, and that they will accumulate at the same rate over time, right? And that would be the substitution rate.
So you have a predictable rate of accumulation. You can
Yes.
use that. Now that rate, it's when you do the math for real, it gets really complicated because it's not actually a linear accumulation. It's according to a curve, depending on how
Yep.
far back in time you go. There's really fun math that goes along
Right.
with it.
So one thing, obviously, is that when you look at, say, a sequence of a particular gene or a stretch of DNA or the whole genome, you're going to see these differences, the mutations that are different between one population versus another population, the substitutions. That will, the closer they are, the closer the last common ancestor there should be fewer of those if there's a consistent rate of mutation, right? And that is a pattern that we see across all animal taxa, all plant taxa, all life. And the thing, though, is that then to figure out the timing involved, some of this then gets rooted down with fossil evidence, right? So a traditional way of doing phylogeny using molecular methods is that they have to have these nodes that are established through fossil evidence that we have. And so some of that, I get the feeling that this is probably where creationists sort of lose their heads, because they're going to see this as sort of some sort of circular
Right.
logic reasoning. But
They
the thing is, we see
definitely
highly
do.
consistent patterns. So if you just talk about, this is an older paper from 1976. I think that some of this has been revised and changed since then. But basically, like looking at, I think in this case, they were looking at 13 different genes. Oh, no, 17 pairs. 17 pairs of a mammalian species from 7 different gene products. Okay. I got it written right on the slide. I should just read the slide. And so if you look at these nodes, that's what this would represent. So this would be like, 2 pairs of animals where we think the last common ancestor was something on the order of around 8 million years ago. And then here would be something like 30 some million years ago. This would be 50 million years ago. But if you just kind of look at nucleotide substitutions in these 7 gene products, and you have some sort of basic estimate for time, this is not rooted in fossil evidence in this particular study. But then you also look at the fossil evidence, you see this nice, clean correlation. And this provides some of the justification for why we can use fossil evidence to then root the molecular clocks. Now, that's my understanding. Please clarify if I have that wrong, or if
No,
there's more that you can add to that.
I think that was excellent. And I love
Okay.
that you picked this figure. I actually use, I think this figure was used in a textbook, and I use a version of it in one of my summer classes, which is an evolution class, where it actually tells you what each point is. So it tells you the species
Oh,
pairs.
right.
And it's great. And I think you're exactly right that once you start saying, oh, and we can correlate this with the fossil record, that's where a lot of creationists are going to want to throw a flag. But here's the thing, the fossils are dated in absolute terms using
Right.
radiometric dating
Exactly.
based on the formations in which they are found or the formations where they are in between. So we can bracket where that fossil is found temporally in the past based on these absolute dating methods. And creationists, I'll stop you right there. I don't want to hear it, because the Oklo natural nuclear reactors exist. They go back like 2 billion years to Central Africa, Western Africa, and they show that all the parameters related to things like nuclear decay, nuclear fission, neutron capture, all these constants that radiometric dating is based on, the Oklo formation shows, it proves, as much as you can prove anything outside of mathematics, it proves that radiometric dating is reliable going back 2 billion years.
Yeah.
So like
Right.
we're looking at a time frame here of 125 million years. Spot on. Those dates are taken to the bank.
Absolutely. Yes.
And the fact that the molecular data correlates so closely with what is essentially radiometric data tells you that the molecular data is extremely reliable.
Yes, extremely reliable. I mean, to see a correlation like this in any kind of data set, especially you're talking about things that are so disparate, nucleotide substitutions in 7 gene pairs in different groups of animals compared to the last common ancestor of the fossils for those pairs from so long ago. I mean, come on. Like, this is clearly a very reliable method. And of course it makes sense since we use genetic comparisons. If we compare genetic sequence information, that's how we know, say, parent offspring relationships within humans. If you use 23andMe, it's the same thing. The fact that I did 23andMe and that they found a sequence that showed that I'm descended from one of the persons on the Mayflower. Wow, great. Of course, right? Like the reason why we know that is because of population genetics and knowing like where these populations have moved and diverged over time, and all of that though they can find out just simply from the sequence of my DNA, and that's true for anyone.
These are fundamentally the same techniques. And a question that I always love when people ask this of creationists is, okay, we accept that you can go back in time and find the common ancestors among all people or all dogs or a bunch of third cousins once removed, right?
Yeah.
And the question is, okay, where do those techniques stop working in the past, right? How far back do you have to go before they stop working? And you point to data like this, and you could add to this data set and you could have different breeds of dogs on there. You could have different breeds of cats on there and say, okay, tell me where the line is where it no longer works.
Well,
and the answer is there is no line.
That's the crux of why I picked the carnivores. And I'm going to kind of highlight that at the end.
Yeah.
That's exactly right. So, okay, molecular clocks, how do they work? So a typical molecular clock is going to be calibrated using fossil evidence. And that the way you can find a substitution rate, this is one way, is to look at overall genetic distance divided by the divergence time based on fossil evidence. Is that correct?
That's the basic way. Yeah, it gets like, if you're going to do this for real, like my background in this is viral phylogenetics. So like when I was in grad school, my work was on base specific mutation rates. So like at what rate does a cytosine mutate relative to, thymine or guanine, and they're different, right? So you have to account for that in your phylogenetic models. And what my colleagues were doing is then taking those data and then using those as the parameters to figure out like the evolutionary histories of different groups of viruses. And when you're looking at viruses are kind of a little bit weird because obviously huge population, short generation times, fast mutation rates, and a lot of variation in the population growth, because if they're in a new population, you get the boom, big exponential growth. But if the population is kind of already saturated, then it's more constant, and that affects your rate of accumulation as well. So there's a lot of other things going on under the hood, but as kind of a first approximation, this is the basic way you're going to do it. Yeah.
Right. And you also pointed to a paper I published like 15 years ago, I think it was.
2009. Yeah.
Yeah. Right. So showing that that selection can have an enormous impact on overall mutation rates as well, right?
Yeah, so because the more selection you have, and in particular what's called purifying selection, basically that's selection to keep a sequence the same. So you can have mutations, but if you deviate from the sequence as it exists, if that causes a problem, then selection is going to reduce the rate at which those mutations will achieve fixation that we talked about earlier. So you have a bigger divergence between the mutation rate, just the rate at which mutations occur, and the long-term rate at which they'll accumulate over
Yes.
many, many generations. And I love that paper from 2009 because a lot of the examples the authors use to kind of check their work, they use the phrase calibrate, which creationists always jump on. It's like, oh, they're calibrating, they're not using the
I
raw
know.
data. No, what those authors do is they calculate what they expect the divergence to be, and then they check their work against human populations with known divergence times. So, like, for example, the Canary Islands and mainland Africa, right? We
Gotcha.
know archaeologically and historically when the Canary Islands were settled and from where they were settled. So we
Right.
can look at the divergence between those populations and say, does that line up with what we expect? And the answer is yes. And the
Gotcha.
nice
Yes.
thing about that example, and they use some Pacific Islands also as examples,
Mhm.
is all of those examples are within the young Earth timeline. So
Yes.
we're not
Exactly.
arguing about things that happened 100,000 or 300,000 years ago. It's within the last 4 or 5,000 years.
Right.
And even in that short little time frame, the rate of accumulation is so slow, it completely precludes the young Earth
Obviously. Yes.
story.
Right. And that's part of what I'm planning on arguing on Saturday, actually,
There you go.
at least as far as the carnivores go. So, okay. There are other things that we got to consider with as far as molecular clocks go. Not all DNA is the same. Mitochondrial DNA versus nuclear DNA. Obviously, there are different mutation rates that occur since, like, nuclear DNA is going to have some more of the error correction mechanisms that mitochondrial DNA does not. I believe that means that mitochondrial DNA obviously has a higher mutation rate and therefore a higher substitution rate. And that is going to mean that sometimes mitochondrial DNA is really well suited for looking at, say, more recent divergence times, whereas nuclear DNA, well, again, it depends upon which gene you're looking at, if there's been positive selection, or these things which we'll get to, ultra conserved elements or regions. Also, kind of getting at what you were talking about last night, that different regions of DNA will have mutations and therefore different rates of substitution depending upon, like, what part of the region of DNA you're talking about. There's a lot of variation that occurs depending on what kind of DNA you're talking about,
Right.
right?
And it could
And
be
then, yeah,
something and it could be something as specific as if you have a gene that is highly active, right? So that DNA spends a lot
yeah,
of time single-stranded. Well, you get a lot of mutations on the non-template strand because when you
mhm,
open it up and you've got enzymes working on this side of it, and this side is just kind of hanging out more frequently than most other parts of the genome, that single-stranded region is just less stable inherently
sure,
when
right,
it compared to when it's double stranded. So like you get these really fine grained differences in mutation rate across the genome based on what the actual physical conformation of the DNA is, how active it is, what it's doing at any given time, and even which strand of the DNA you're looking at can have different mutation rates based on what each strand is doing, a higher frequency or a lower frequency of the time. It's
sure.
really fun digging into that stuff, and it leads to some really weird patterns.
Yeah, right. And that's kind of basically what you were talking about last night with the Aridopsis
Exactly.
study from 2022, I think is when that was published, right?
Yes. And
Yeah.
that's
So,
that's the one that has within the Arabidopsis genome, you can have regions that have a higher or lower inherent kind of
yes,
mutation rate. Now,
right.
there's a lot of pushback against that paper, and I think a lot of it's valid. And I didn't go into that last night, so there's a little
Mm,
disclaimer there. But
okay.
what the paper shows is that you have region-specific mutation rates within the genome. Yeah.
Yes, right. So the other thing is that calibration is an important aspect. You already talked about some of the calibration that's done, with the Canary Islands versus other areas. Ancient DNA is something that can be used to
Yep.
confirm substitution rates, and that's something that we see. It's new technology where you can extract DNA from fossils and from the descendants of, say, Neanderthal versus human, and you can look to see, do we see less divergence in the ancient forms versus more recent forms, that kind of thing. Like, they should be closer if you look at more ancient forms. Isn't that basically right?
The more ancient ones should be more similar to each other compared
That's
to
right.
like the more right. So you have
Yes,
Neanderthal and then like preserved human remains. We'd expect to be more similar
exactly.
compared to Neanderthal and modern humans.
Like this. Basically, if you could go back
Yeah,
and look at the
right.
DNA of the ancestors of two different forms, they should be more similar to each other than if you're looking more recently. And I think that this has been shown in Neanderthals and
Yeah, right. So if
yeah,
you're looking at, a human preserved specimen from
right,
45,000 years ago and a Neanderthal from 45,000 years ago compared to the Neanderthal and a human from right now,
right,
right, the human from right now should be more
more
divergent because
divergent.
there's been
Yes.
more time
And that's
for
something that
exactly.
we've
Yeah.
seen, confirming again that like this genetic
Predictions.
equidistance actually works because you can actually go back and take a look at see what the sequence was 50,000 years ago or something like that. So it's really cool. I've been trying to find some of these basic kinds of comparisons that have been done in carnivores. I found one interesting study that I kind of hope to bring up on Saturday, but we'll see. It kind of
By the
depends on
way,
the conversation.
before we
Yeah.
move on from that point,
Yeah.
that because the Neanderthal thing is so beautiful
Mhm.
because it is crystal clear, and we now have both the mitochondrial DNA and the nuclear
Right.
genome. So it's not just like one little thing; it's a lot of comparisons that we can do. And creationists, the most famous creationists that deal with this kind of thing, have a really, I think they've got a really crafty way of dealing with Neanderthals and figuring out how to kind of work Neanderthal ancient DNA into their models. And here's the secret, folks. This is Nathaniel Jeanson's trick for dealing with Neanderthals. He pretends they don't exist.
Yes, of course. Yeah.
That's
Of
the
course
way
they
they
want to
handle
ignore
it.
it. Yeah.
They just wrote a whole book on this. Didn't even mention it. Just
Not even mentioned
doesn't
at
even
all.
come up
That's
once.
of
Yeah.
course. Yes. No. That's actually, one of the things that creationism does that's consistent with other similar sort of world warping reality warping worldviews, which is that it's totally built upon confirmation bias, that you will highlight anything that supports your model and you will completely ignore all the other stuff that is totally inconsistent with your model. They do no work whatsoever to try to incorporate all the data into the
Nope.
world model. Yes.
Nope.
So coalescent models using genetic diversity in populations to estimate substitution rates. This is something I took from some piece that I was reading someplace. I actually don't fully understand what this means. Do you have anything to say about this?
So let's see. We use genetic diversity.
Coalescent, yeah, genetic diversity in populations to estimate substitution rates.
Yeah, so what we're looking at here is it's kind of what we talked about with the calibration with like the Canary Islands, for example.
Yeah.
So
Okay.
like you're looking at
Ah.
modern genetic diversity in the two
Sure.
populations to say, okay, they shared a common ancestor. I
Yeah.
forget what it is for the Canary, 2000 years ago, whatever it is, right?
Mhm.
They shared a common ancestor at this point. Since then, they've accumulated a certain number of
Yeah.
differences. Now we have that common ancestor time locked in. We know when that was. So now we know the rate of accumulation since then.
Right. Okay. That makes sense. Comparative genomics in many different ways. Obviously, this gets even better with full genome sequencing. The paper that the figure that you talked about as being part of your lecture, where they're comparing the fossil dates to nucleotide substitutions. That's back when, trying to figure out the sequences of things was a very laborious thing. They did 7 genes across, many different mammalian pairs. I imagine that was a huge amount of work that went into that. Nowadays, all you would have to do is just look it up on the NCBI database, and you could have the answer to all of that done in minutes. And if there were, or maybe it would take more than minutes, maybe a couple of hours of work, but and you could do that for thousands of genes, really, to get all that done within a few days if you wanted to. And then if you needed the sequence of something, well, you can work with your local sequencing center and have that data within days to weeks and you could have the entire genome of a whole new species that would be there. Now, obviously, you got to go through the annotation process and figuring out which genes are which.
Right.
But because we have so much, that takes a little bit more time.
Oh yeah.
But we have lots of scaffolds to work with on that, and our understanding of how genomes get structured just gets better and better over time. And all of that is consistent with the phylogeny that we understand across all animals.
Right.
That's all I really got to say about comparative genomics.
It's
Last
really
thing, yeah,
it's
go ahead.
really important to note here that as you
Yeah,
talk about just the explosion of data that we've collected on this question over the last, figure 20 years, right?
yeah,
Once
20
sequencing's
years.
gotten just way cheaper, way faster. I
Exactly.
mean, again, in grad school, I was doing this with viruses, but I would send out dozens of samples of viruses at a time. And 2 days later, I'd have, a bunch of genome sequences in my inbox, right?
Yes.
And that just wasn't possible 10 years earlier, even for
Right.
those tiny little genomes.
Yeah.
And the important thing when we're talking about all of this in the context of creationism is that every time we do that, that is a test of the evolutionary hypothesis,
Yeah,
right? Every genome we sequence, that is a test of the evolutionary
it is.
hypothesis. Does that genome fit where we expect it to in the context of all the other members of its population, of its species, of whatever clade you want to talk about, or does it look very different in some way?
That's
And we have never found, to use an expression, right? We've never found the Precambrian rabbit, right? We've never
yeah,
found that in the genome. It's
that's right.
always been consistent with the nested hierarchical pattern that we established very early in the enterprise.
Exactly. Like when this was all getting done in its infancy in the 70s and the 80s, it was Herculean amount of work to just get the sequence of, say, a single gene, let alone 7 genes. Um, but they did it, and it was completely consistent with a hierarchical pattern of phylogenetic relationships with common descent. And but now every time that we add an entire genome, all of it is fully consistent. We see the same exact pattern over and over and over again with every single genome that's been sequenced. So, and we add more genomes every single day. So the last thing, and this kind of brings me to the last point, which is really the crux of the paper that we're talking about pedigree studies. So this is measuring mutation rates directly in parents and offspring, right? So this is the paper, right? This was published in Nature a couple of years ago. I wanted to include all the institutions that are involved with putting this paper together. This was a huge project. So this is the list of authors, and you can see it goes out to like what is that 17 different institutions. And yeah, like this is the thing about collaborative work like this gets done across the entire world. We got institutes here in the Americas and Europe and in Asia. I mean, it's a big project, and that's kind of what you would need to take a project like this. So let's get into it. The title of the article is Evolution of the Germline Mutation Rate Across Vertebrates. Okay, so what did they do? They went ahead and sequenced the entire genomes of 151 parent offspring trios from 68 vertebrate species. So what does that mean, parent offspring trios? They did the sequence of the father, they did the sequence of the mother, and they did a sequence of an offspring of that parent. And so that's the trio. And they did that 151 times. It's a lot of genomes to look through. And they did this across 68 different vertebrate species. So with that information, they can then estimate the germline mutation rates across different taxa. So basically what happens here is that if you have a mother and a father, and you can know what their sequence is, and then if you find new mutations in the offspring, within that offspring, that means that offspring had some sort of germline mutation because it didn't exist in the parents. Do I have that right?
Yeah.
Or am
So
I not
when
saying
we
that right?
I think a really important thing to make sure
Yeah,
everyone's clear on is why
please.
we're specifying germline mutations,
Yes,
because
right. I didn't
mutations
make that clear.
occur, just your DNA just sits there and it'll spontaneously mutate. And also when you copy your DNA, most mutations are due to copying errors. And then we
Right.
have really good proofreading that catches most of those errors, but some of them slip through. Now, the lining of your stomach is completely replaced about once every 3 days, right? So those cells are replicating really fast. They're making copies of their DNA, and they're introducing a lot of errors, a lot of mutations in the lining of your stomach compared to like your muscles, your skin, for example. But those mutations are never going to make it into your offspring. The only mutations that are ever going to make it to your offspring are mutations that occur in the germline, the cells that specialize into either egg or sperm. None of the other mutations matter. And all the other mutations, we call those somatic mutations, those do not matter for evolution because they're never going to get passed on. So, like a really simple example of this is you get a bad sunburn, right? The UV radiation in your skin causes mutations in your skin.
Yep.
Well, your kids are not going to inherit those mutations in your skin because you forgot to sunscreen one of your arms, right? You fell asleep on the beach. Those mutations are in your skin cells. They're not getting passed on to your offspring. So when we talk about the kind of one generation to the next mutation rate, we have to specify the germline mutation rate because those are the only mutations that can be inherited by the next generations. That's an important caveat for everybody.
Yeah.
We're not talking about all the mutations that occur. We're talking about a teeny fraction of them that occur in the germline.
Exactly. And that's a very important point. Exactly. And that when this occurs, it would have to happen very early on in development. So, obviously, when a mommy and a daddy love each other very much, sperm meets egg, then you have a fertilization that occurs. That's when there's now an offspring. That cell will divide into 2 cells, 4 cells. Eventually, you're going to get specialization of cells as they develop. As you said, most of the cells will end up being somatic, but some of them will be part of the germline. The mutation has to happen either within the germline or prior to the emergence of the germline, the specialization of the germline in the developing embryo. As you said, any sort of mutation that gets accumulated outside of the germline, outside, within the somatic parts of the body, it doesn't matter. It really, not at all. You can be totally irradiated on the outside of your body and everything being changed. If it isn't getting into the bits, that doesn't, it won't help you there. So right. And it won't be passed on to the next generation. That's the thing.
Yeah.
Yes. And that's what obviously is what's going to be important for evolution.
Yeah.
So,
by the way, creationists love this pedigree mutation rate, because there are a couple of papers. I know you know exactly.
I know. No, I don't actually, because you mentioned that in our in your email, and I was like, really? Because there's a lot of things that actually work against them.
Let's put it this way. They think they like the pedigree mutation rate based on a couple of papers, some of them from as early as 1997 or 1998. And so what creationists say is that you look at this one generation rate, parent to offspring, and you calculate that a lot of times, you're going to get a big margin of error. And for some of the studies, that margin of error on the fast end, the rate of mutation at the fast side of that margin of error, gets you to where if you take that fastest rate and just do the simple multiplication, mutations per generation divided by the number, you just do the simple math, you can squeeze all, for example, the mitochondrial variation into 6,500 years.
Okay.
Now, that's on one end of the margin of error. There's the rest of the range.
Yes,
obviously. Also, those tend to be the earlier studies with smaller data sets. And the newer studies, including studies that span not just parent offspring, but also have cousins and go back, instead of just to one set of parents, they'll go back multiple generations. You use the pedigree rates from those, and it's the evolutionary rate. It's the longer term rate.
Much longer term. And I'd seen that other studies have shown that the substitution rate that we get from pedigree studies, it's actually a bit slower, or at least lower than what had been used as some molecular dating methods, in humans. And when we're talking about nuclear DNA, that required some revision of some of the dating, and that maybe our fossil nodes aren't actually matching up quite well, because some of those pedigree rates, when you're talking about nuclear DNA, they're almost always consistent totally, but some of them are actually quite slow.
Yeah.
Right. And that's going in the opposite direction of where creationists would need it.
Right. They're wrong in the wrong way for creationists. Exactly. Where creationists could say, oh, these numbers are wrong. And you go, okay, do the math. And the number gets worse for them. It's
worse for them. Yes. And so when you said that they are, they're big on, I didn't realize that they're taking pedigree studies based on mutational rates out of mitochondria. Okay.
Oh, yeah.
Sure. Yeah.
Oh yeah, it's a paper from 1997, 1998. It's the Parsons paper, and they love to talk about how the FBI uses that rate for forensics for so if they're looking at the sample and the number of differences in the sample, and it's think for 5 seconds about why that's applicable for forensics and not phylogenetics. Think for 5 seconds why that would be the case, guys.
Yeah, exactly.
Yeah.
So, all right, the other thing that they did, so they have all this information about mutation rates across these studies, across these different species, and using these trios. And they can then compare them to other things that we know about these species, such as looking at life history things, generation time, age at maturity, and then compare them across the different groups: mammals, birds, reptiles. Do we see any kind of patterns? So, I think that there's this study, it's published in Nature. Obviously, we're supposed to bow down to anything published in Nature. And I'm not being too facetious here. Like, obviously, it's great work that they did, and I don't see any reason to not believe it. But there's like, all right, they did a ton of work, and then they end up doing some correlations and things. So in some ways, it's a little disappointing. But
Yeah,
we'll get there. Yeah.
I agree that there's
Yeah.
the kind of analysis, I think, is less impressive than the data set,
Yes. And
right?
this is the data set,
Yeah,
basically.
this is
Yeah.
really cool.
This is very cool.
This is, and for
So,
our
yeah.
purposes, this is very cool.
Absolutely. For our purposes, it's very cool. It's very cool just overall. So obviously, this is on the left. This is the phylogeny of the different species that they include in their data set. This is one that they generated using their sequencing data. The different numbers here represent fossil nodes. So they use basically fossil-based methods of rooting some of this. But, like that's all very consistent with how molecular clocks are done. And they got it into 4 different basic groups. So they didn't include amphibians, which of course bugs me since my lab works with amphibians. But we've got fishes here. They've got a bunch of different mammals. That's the orange. They've got reptiles and they've got birds. And their phylogeny matches every other molecular phylogeny that's ever been published. On the right, this is the part that's really cool, because this is now showing the pedigree-based mutation rate per site per generation. And now in the paper, they talk about how the range is something about 40-fold range. And, yes, it's true, but it's because of just one kind of outlier, which obviously you can see that so this is the rheas. They're, the flightless birds, ostriches are another example of these. The radites are kind of this weird group of birds that are the hagfish of all the birds. You can see that they're the one outlier group compared to the rest of the clade. They're not even included in aves necessarily. Well, they are aves, but they're not neo-aves, right? So because they're probably from a lineage that pre-existed the death of all the dinosaurs. So they go back quite some time. That split. And we have right, they're one of the outliers, but mostly what we see is the pedigree-based mutation rate is something on the order of around 1 times 10 to the negative 8, which is fully consistent with the substitution rates that we see from using these other methods. Isn't that your understanding?
Yeah, it's the fact that they have such a big data set is really nice here.
Yes.
And even where there is variation, I mean, that variation, just looking at, I like to look at the mammals because that is the largest kind of subgroup here in terms of the number of species. And you can see that is a fairly tight band from, 0.4, 0.5 times 10 to the negative 8th, all the way up to about 1.5, right? It's a fairly narrow band in there where we find those rates, which is, should give people confidence that this is not, spurious, that these are fairly reliable numbers.
Yes. I mean, we're talking about, 0.2 to about 2 as the full range. So that's just one order of magnitude for 36 different mammal species. It's pretty great. And basically all the other species are within that range as well, except for a couple of outliers.
Yeah, a couple of weird ones, but
yeah, just a couple of weird ones. Yes. And that's totally fine. So, right. This is basically the sum total of like the major corpus of what they did here. There are a few other things that they do. Do you want to say anything else?
Yeah, just the key thing
please.
with this is, because I think in the chat here, our friend Mike, Answers in Atheism, is going to say, Hovind is going to say this is just drawing, this is just a page, just lines on a page, right? They're just making this up, right? But like all of these data are directly gathered, directly observed pedigree data. And what you care about for the purposes of this debate, right, is the rate. The lines, the phylogeny on the side of this figure is kind of beside the point. Right. What we care about is the rate. Yes. Those rates, those are not just like inferred, those are directly measured.
Directly measured. Yes,
exactly. And the important thing for creationists is that, and when we're having this conversation in terms of how long it would take for certain things to evolve or whatever, right? This rate that we're seeing right here for creationists, this is your speed limit, right? You can have a substitution rate that's slower than your mutation rate, but it can't be faster, That's
That's
not
right.
possible. So
Yes.
whatever amount of variation you have to explain given an observed mutation rate, these are the numbers you have to work with. And if these numbers say it would take 50,000 years or 100,000 years or a million years, well, you're hosed because that's your speed limit. And we've directly measured it.
Yes, and their numbers, I mean, in order to get it to work with their model, I mean, we'll get to this, but they'd have to be somewhere on the order of around 5,000 times higher,
Yeah.
and they're not.
Yeah. And it's right.
So,
It's not
yeah,
close. It's
yes,
orders of magnitude
right. Even
difference.
if you account for the full range of being 40-fold, just allowing for the radites, down to whatever this guy is, uh, it still is well below 5,000. So, right.
Yeah.
Kind of host. So there are a few other things that they do. They and some interesting things that pop out of it. One of the main kind of results that they find is that overall parental age at reproduction is correlated with this mutation rate that they see. And some of that just makes sense. And this was predicted anyway, that species with longer generation intervals should have higher per generation mutation rates because the longer you live, you're going to have more cell divisions, particularly in spermatogenesis, as we'll get to in a moment, and more time for DNA damage to accumulate, right?
There you go.
That makes sense.
Makes
And
sense.
this is perfectly correlated with that. The other thing that they show is that GMR is germline mutation rate, that it has a male bias in general. So other than for the fishes and for the reptiles, basically the male bias, so the more that these bars are towards the right, means that there were more mutation rates that they could ascribe to having occurred within the parental contribution to the trio than the maternal. Sorry, the father than the mother, and paternal versus maternal. I think I said parental. So
And that
basically, yeah,
Yeah, and that makes perfect sense
yes,
given the differential
explain.
parental investment, right? You look
Yes.
at birds, and I bet if you had amphibians on here, I bet amphibians would be more like the fish on here with relatively
Mhm.
small differences
Yeah.
because the difference in terms of the energetic investment in the gametes between the two sexes is much smaller. Whereas you think mammals, you think birds, right? The female investment is much larger, which means you have fewer gametes per unit time,
Yep.
which means fewer cell divisions, which means
Yep.
less mutations.
That's
Whereas
exactly right.
males are just making sperm the whole time anyway. So
Yep.
you just have lots of mutations occurring in those germline cells that specialize into sperm. So these data, this is great. This is exactly what we would expect
Yep.
based on the life histories and the ecologies of these different species. It's very
Exactly.
cool finding.
Yes. Basically, the reproductive ecology. Yes. Um. I don't know if this is terribly important. We may skip over this, but I wanted to get your impression on their what was it? Ultra conserved element substitution rate versus whole genome analysis substitution rate. And they find that there's essentially a correlation now. I think this is a little bit spurious because it's being driven by like a few outliers mostly.
Yeah, it's a lot, right? It's a cluster with then a
It's a cluster.
few other
Yes.
things.
I always hate correlations like this, so I don't wanna take too much from this,
Yeah, I
but you can
know.
always kinda like, if you narrow it to this, you can kinda see some sort of
You can
relationship.
see there's like a
Yeah,
little,
sure.
I can,
But
I can
anyway, ultra conserved elements. Now, if you wanna talk about, like, how does that relate to, um, I guess a little bit of what you talked about in your video that you released last night?
so you're
It doesn't. Mhm, go
So when we
ahead.
talk about conserve, like evolutionary
Yes.
conservation, just means
Right.
it tends to stay the same over
Mhm.
long periods of time. So you look at parts of the genome that are really important. If you mess with them, things go bad really fast.
Mhm.
Those regions, it's not that they don't experience mutations. There's some species where, like we talked about last night, like maybe, but you could assume that even if they experience mutations at the same rate as the rest of the genome, those mutations are much more likely to be harmful and therefore selected out of the population. If you have a mutation, for example, a good example of this is the ribosomal RNA, right? It's
Mhm.
the core of the ribosome that's involved in protein synthesis.
Yeah.
So it's
If you have a
DNA,
mutation there, you can't really make any proteins,
then your
and
ribosomes
yeah.
don't work, right? So if
Yes.
you look at the ribosomes, it's really hard, for example, to resolve a phylogenetic tree of ribosomes within the primates because they're all basically identical.
I see.
Even
Yes.
across all those millions of years,
Right.
they're so tightly constrained that the rate of substitution in those regions is very low. But you
Yeah.
compare that to other regions of the genome, that don't have that level of constraint, even functional regions, even, protein coding regions,
Right.
you're going to have a higher rate of mutation accumulation just because they're not as set in stone. It's not as critical that they don't change at all in order to maintain that function at a very high level.
And the
And
thing
so
is, like, if I could interject, I think that I mean, my impression is that, like, of these genes that are ultra conserved, like, what percentage of, say, all genes would you say are like, and I know this is getting out of your area of expertise, but like within humans, for instance, how many of those are considered would you consider to be like ultra conserved?
I wouldn't even want to speculate because I
Okay.
do not know the answer. I can give you a
Yeah.
couple of examples, like ribosomes are
Ribosomal?
like the
Sure.
bit,
That makes
you
sense.
can compare ribosomes across domains, right?
Yeah.
Bacteria, archaea, eukarya, you
That's
can
right.
resolve a
That's
phylogeny.
why we use it. Yes, in
That's
fact,
how
that was one of the
consistent they are.
reasons how we are able to resolve archaea are different from bacteria is because of
Exactly.
ribosomal RNA.
Another
Yes.
good example is, which I actually think is the example in the that figure that you showed earlier with the
Mm,
millions of years and the number of differences with the line. That one is cytochrome. I think that one uses cytochrome C oxidase, which is a protein that's involved in cellular respiration.
mm.
And so that's a very important kind of energy shuffling
Yes. Yep.
component. If that thing doesn't work, you're going to have problems. And so
Yep.
that is remarkably consistent
Right.
across lots of different groups. Um, I wouldn't want to speculate on any other ones because
That's fine.
I'd
Yes.
just be speculating. But
You'd be speculating. Yeah.
those are two that I know of that are very
Very
consistent.
ultra
That you
conserved.
can make
Yes.
comparisons across extremely divergent groups and still
Yeah.
get useful information.
And at this point, because we have so many whole genome sequences, we could start to create a database of like just how highly conserved, some genes are relative to others. One of the problems, of course, is that our, it's one thing to figure out if something is a gene. Like we have programs that'll just tell you this is a gene. And a lot of times, we can figure out, oh, it's most similar to this gene. So therefore it's part of this gene family.
Mhm.
There's within humans, I would say, and vertebrates, genetics in general, like we have a reasonably good understanding of about like half of all the genes and like what they do and where they interact and that kind of thing. The other half, I would say half of that again is probably just estimates
It's like,
based on modeling.
it's
Yes,
got it,
like, oh, it's in this gene family. It
right.
probably does this, but we don't really know,
Yeah,
and no one has actually studied this. The
right.
thing is, like, there's around 30 some thousand genes essentially that create proteins within the human genome and basically all vertebrate genomes. And yeah, like I said, we kind of know about half of them, and I don't even mean like we know them very, very well. Like,
We can
oh,
identify like the
yes,
protein
we can
family or
identify
something.
the
We know what it
protein.
kind of is.
Some work has been done, like it's shown up in some transcriptomic type analysis, and therefore it seems to be related to some process. But like about half of them, we don't know a whole lot about.
Right.
So there's still a lot that we don't know.
There are some that are, we could say, we could look at it and go, okay, we've got a promoter, we've got a start codon,
Right.
we've got a stop codon, we've got a
Mhm.
transcription termination sequence, and we know that it's transcribed and it's
Yes,
translated.
and
And
that's
that's
it.
therefore it's a gene, and that's
Yeah.
what we got.
Right.
Yeah.
Yes. Because it, to figure out what it does requires actually now like a considerable amount of work. There's only so much that can be done in silico. You have to get into some cell or an organism and start playing with it and modifying it and
Yep.
measuring it in order to really understand what it does. And
Yep.
yeah, we're just limited in how much we can do.
And a bunch of them you've got what number did you say for the number of genes? Because you've got
That's
like
around 30,000 genes.
and
Yeah,
uh,
estimates from
is that all
25 to
protein
30.
coding? Because I think because you've
That's
got a
protein
whole
coding.
because you've got a
Yes,
whole
because
bunch of RNA genes as
yes,
well,
it's
right?
not including those.
Right. So you've got your protein coding genes, you've got your RNA genes,
Right.
and then so the RNA genes, there's a lot of those where we've identified, okay, this thing is highly transcribed, it's
Right.
highly constrained, right? It's under purifying selection. We
Mhm.
know it's active, and that's what we got.
Yep.
Right?
Yes.
There's a lot of
That's
that
it.
piece of RNA floating around.
That's
It's
right.
it's energetically demanding to
Mhm.
have that piece of RNA floating around.
Mhm.
And if you mutate it, that comes with a cost. So we know
Right.
it's doing something. mm,
Yes.
And here we
Yes,
are.
right, and so for some of them, we know that it's doing something. One thing about the the RNA, creating genes that don't result in any sort of protein, they're not quite as energetically costly as, say,
Right.
generating the protein, right? And
Right.
that's one of the things that can
Really good
allow
paper on
for,
that
yes, right. It's
from
quite a
Lynch.
bit lower than that. Now,
Yeah.
if you're generating a lot of
A
RNA,
lot of
like,
it.
it's
Yeah.
dependent upon how much, then it starts to become energetically costly. And then obviously if you have a lot of something, well, and if you break it and then it does something to the organism, well, that clearly indicates that it's playing some sort of role. But just because we have things that generate RNA doesn't necessarily mean that it's actually doing much,
Yeah.
right? As we know from
Not even
ENCODE,
close.
and yes,
Don't
don't
even
don't
get me
go there.
started on
Yes, I
encode.
know. I
Don't even
know.
get me started.
I have big problems with ENCODE as well. So, all right. The other thing one other thing that they highlighted is that genetic mutation, germline mutation rate was associated with the number of offspring. They find that if you have fewer offspring in all species, that you have a slightly higher mutation rate than the ones that have many. They found that this is particularly significant in mammals, and there's only sort of a trend in birds. But the other thing that they say in the paper is that, well, this is actually probably because animals that have longer generation times also have fewer offspring. So therefore, this is just a thing
Right.
that's correlated with a thing, and it's not. It's a series of correlations. So this is
Right.
one of the places where I started to read this paper and be like, all right, we're kind of digging here for,
It's
yes,
cool, but it's a lot of what you would expect based on like
yes,
life history trait evolution.
exactly,
And
yes,
you have when you talk about life history, folks, we're talking about things like how long is your lifespan? How many times do you reproduce? How many kids do you have? How much energy, how much like parental investment do you put into your kids? How many kids do you have at a single time, right? These are all life history traits. And a lot of that correlates together such that you have kind of two broad strategies. You have two extremes, the R strategists and the K strategists. But even beyond the extremes, you have kind of two broad strategies where you could have relatively few kids and put a lot of energy into them and maximize their survival. Or you could have a lot of kids and put very little energy and just play the numbers and, good luck. And maybe 1% of them survive and that's enough.
right.
And this correlation is kind of what you would expect based on what we know about those kinds of life
Yep.
history trends, which is
Given that
cool.
we also know that generation time tends to be associated with that R selected versus K selected
Yeah.
distinction that we find in animals. And they already showed that generation time is increased, or that it's correlated with an increase in germline mutation rate. Therefore, fewer many. Okay, so they
And
also see another correlation.
and a fun kind of cross correlation here that's really nice is mammals tend to be more on that few offspring, put a lot of energy into them thing.
Right.
Things like fish are way in the other direction, right? Think about Finding Nemo, right? And then birds are kind of in the middle. And
Mhm.
that's what we see right here,
We
where they're
see it as
like,
well.
it's a
Yes.
trend, but it's
Right.
not super strong, right? It is
Yes.
really nice to see basically that same kind of continuum of
Yep.
traits, just in a different context here, but it's showing basically consistent with
It's a
this
sim
kind of
with
trends in life histories.
exactly what you would expect. And that's
Yeah.
great. Good that it's consistent. It would be interesting, or maybe even scary if it wasn't. Not surprising. That's the one where my disappointment was like, okay, well, obviously these things are going to be
Yeah.
correlated. But as you point out, like the mammals, as you say, generally they tend to have fewer, especially a lot of ones that they include. But there are several that they include that have many, and they spend a lot of time talking about the rodents, which
Mhm.
have a lower mutation rate per generation.
Which
So
is
yes, which is
perfectly
fully consistent.
consistent. Again,
Yes.
consistent with what we expect, but again, the data set itself is more interesting than the
Right.
analysis, right? It's like,
Yes.
yeah,
So the last thing that they talk about is this thing that they find
this
with
is cool.
domesticated species. Yeah, so they find that yearly germline mutation rates are actually higher in domesticated species than non-domesticated species. And but then they redo the analysis and B. I'm just going to ignore that for now because it's not necessarily terribly important to the point that we're making. But the on in A, they this is where they're doing their basic analysis and they have domestication status versus not. So animals are domesticated versus not. And they find that the domesticated versions tend to have a higher mutation rate versus non domesticated species. What they argue here is that there's strong selection, artificial selection in this case, to have shorter generation times. And therefore, there's an increase in the mutation rate that occurs.
There you go.
Yeah. So basically, the main take points that I take from this paper is that their measurements for germline mutation rate are consistent with the estimates that, mutation rate, then eventually substitution rates that are reported using other methods. It's fully consistent. And as you said, it kind of puts the speed limit as to what that mutation rate can be. It can't be higher than this, right? And you need a much higher mutation rate in order to make this work um,
Exactly.
for young Earth creationism.
And
Yeah.
and focusing on carnivores specifically, so you could talk about,
Yep.
Felidae, the cats, Canidae, the dogs, Ursidae, the
Yep.
bears. I don't know the other groups within Carnivora, but there's other
We'll
ones you
get
could
there.
talk about. And the great thing is, creationists basically equate each of those families to a created kind.
Yep.
So to get all of the diversity that we see in there, you have to be able to get that from your arc pair 4,500 years ago. We have a speed limit. Can you generate all the variation we see with that speed limit in the amount of time you have?
Right.
But then it's worse because you don't have 4,500 years. You have
Oh,
a couple hundred, you have
yes,
maybe 500 years.
exactly.
Because
Yes.
by the time you get to like Middle Kingdom Egypt, we have direct historical
Yes.
records for like the diversification of the cats, for example, or the
Absolutely. Yes.
waterfowl, or this or that. So
Now,
you
the
don't
cats
have
in particular is where I plan on going because, like, we have Sumerian evidence that, like, the different species of cats were already existed.
Yep.
The Egyptians have it. They have leopards and lions and
Uh-huh.
tigers. Yes, they all existed within a few hundred years.
And you have an ice
And
age in
yes.
between. So you've got
Oh, do they
Ark.
believe that there's an ice
Yeah,
age?
you've got Ark,
Oh, I didn't know
and then
that.
Ice Age, and then Middle
Okay.
Kingdom Egypt, and that whole thing is like 500 years, maybe. So you just don't have. I mean, you're
Uh-huh.
talking for some, like if you're looking at, I don't think that cats are quite this fast, but if you look
Mm.
at like elephants, for example, you literally need to have a speciation event every generation in
Oh,
order
wow.
to, it becomes like you need to have evolution happen the way Kent thinks it happens, where one
Mhm.
thing literally gives birth to a different thing in order to generate the species
Right.
diversity in that amount of time.
Yeah, that's a great way to put that. I might steal that line for
Oh, please.
you. Because that's exactly right. So, and that's, as you said, you don't know all the other carnivore groups. I also have been learning a fair amount over the last couple of weeks. There are, as far as the creationists sort of delineate these things, they generally do it along the order, along families. So they just, this is at least from Answers in Genesis. Erica, who's got a gibbon, she has that great video where she went to the ark, and obviously that's an excellent video, but she also had posted online someplace a list of all the kinds. And
Painstakingly
yes, I
took
I'm
that
so
big
glad
post
she
it.
did that. Yes, so I went through that and I picked out all the carnivore kinds that they include in that list. Now I know that
Beautiful.
Kent does not bother to list any kinds, but I'm going to have to kind of work on what, at least, I mean, that just also illustrates how intellectually bankrupt this whole phenomenon is, that they
Yeah.
can't even bother to come up with kinds. At least Answers in Genesis tried, but all they did is basically list families. But what that means is that the 57 different species of the mustelids are one species. This includes things like badgers and meerkats and mink and that kind of thing, ferrets. There's 57 different species across the world, and this is from this study where they did this kind of molecular phylogeny of just the adaptive radiation, yeah, amongst the mustelids, um, and then some of the outline groups as well. So the procyonids as well they included, which include the raccoon-like animals. Now we think of, oh, raccoons, so they must be all just like raccoons, but they're really quite different.
Right.
They're these, and then the red panda is not a panda. It's actually more closely related to. It's kind of the hagfish of this entire group. And then you have the Aluridae, which are the skunks and those related animals as well. All of these I'm sorry, the Mephidae, which are the skunks. Those are the another carnivore group, also considered a kind. The thing is, so as far as we agree and creationists agree, that there has to be this sort of relationship within a kind, right? They
Right.
think that this would be like the flood, and that there's a phylogeny that would explain, say, all the procyonids that explain that, but they have to jam it all, like you said, within at least within 4,400 years But realistically, especially when you look at things like the cats, for which we have substantial archaeological evidence that they existed 4,000 years ago, that has to be compressed within 500 years,
Yeah.
and it just doesn't work. It
Yeah,
doesn't work at all.
and that's being overly generous because,
Mhm.
we have really good records. Middle Kingdom Egypt, which I just looked it up, starts approximately 2000 BCE. Now, flood consensus among young earthers 4400, right? So you've got 400 years in there, minus, a century-ish for an ice age. Okay, so you've got 300 years of diversification that can happen in there. Also, there's Old Kingdom Egypt.
Yes, right.
We're just giving that one away.
I know. Yes.
There's records that are unbroken
Yeah.
that go back to before the flood.
Before the flood. Yes, we know that. Yes. Which is why some young Earth creationists have started to get a little slippery with that 4,400 year timeline. They
Yeah.
are starting to be like, well, it's more like maybe 5 to 6,000 years
Yeah,
ago.
and then you start hearing about how, well, Usher was only one person that did the
Right.
calculations. Other people
Yes.
calculated 7,000 or 8,000. It starts slipping towards
It starts
10
slipping
in
even
order
more.
to
Yes.
to account for, yeah.
Right.
Yeah, but
It's
based
still,
on,
but even if you were being so generous and you say
oh
that
yeah,
it's 10,000 years ago, none of these data are consistent with that. You
no,
cannot, you would have to have a mutation rate that's around 1,000-fold of what we see, a substitution rate that's around 1,000-fold than what we observe,
it's
to explain any
impossible.
of these phylogenies.
Yeah, it's impossible.
But yes, but all of this is fully consistent with the fossil data and the way that comes out through radiological dating. It all matches up perfectly.
And where
Yeah.
I would hammer Kent on this is
Mhm.
what I would do, and I'm never going to get to do this because I don't think I'm quite
You're
confident
done with him, I
that
hope.
man genuinely detests me. The only time
Yeah,
I get to
me
talk to him
too.
is if I can
Yeah.
ambush him in an open mic, but he doesn't do many of those anymore.
Yeah,
But
I've noticed.
I would make sure the focus is on him being unable to explain this. And
Yeah.
say like, look, you have 400 years to go from A to B given this rate of change. You have to
Right.
explain this much with
Mhm.
this much is what, rate times time. This is what you can do. This is what you have to explain. Reconcile those two things. And I
Yep.
would just hammer him on it. He'll go, oh, you think a tree gave birth to a cow. And then you could just be like, Kent, would you like to answer the question I asked? Would you like to try to make sense? Because your
Yeah.
numbers say that this happened and then this happened, and we've directly observed the rate at which that thing happens, and it
Right.
can't fit. Like,
I
explain
know.
to me how this works. Or you could go back to your script. Like, just
Yeah.
hammer it and spotlight it. And,
Absolutely.
like, don't get sucked into the, like, try to explain science to him because, like,
I know. Yeah.
talking to a brick wall, right? But,
Exactly.
like,
Yeah.
the whole point is your model can't explain this. My
Yes.
model, all the predictions match. Your model can't explain this.
And explain this.
Explain how I'm wrong.
Exactly. That's right. Exactly.
And then he'll go to his script and he'll point out he
And
didn't
go to
even
the
try to
script.
explain it.
He'll talk about how the Excel program and the Word program have 80% similarity, and but that means that there's a common designer. And I actually have responses for that as well. So
Or
you know,
or or he'll say you can't explain the origins of the carnivores if you can't explain the origins of DNA and Earth and stars and blah 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 blah. Right?
hey, and I can say the debate was about evolution of carnivores in
Carnivores.
particular. Talking about carnivores, that's why I made it so specific, so that we
Right.
don't have to go back to the evolution of the universe in his 6 stages of evolution.
You could even
We're talking
say,
about one particular group.
yeah, you could even say up front, Dapper Dinosaur did this to great effect a few years back.
Mhm.
You could say up front, for the purposes of this debate, I am conceding that the universe was created, that the sun was created, that the earth was created. I am conceding all of these things.
Yep.
And we're going to start from the position that animals were created. Now, the thing we're going to argue about is, did that happen millions of years ago, or did that happen 6,500 years ago? And go from there, right?
That's
And
right.
we're going to, for the purposes of this debate, we're going to concede all that other stuff. And
Yeah.
Dapper Dinosaur did this a bunch of years ago in a debate with Kent, where the topic was, are birds dinosaurs? And the way he did this was brilliant, because he started the debate by saying, I am not arguing that birds evolved from dinosaurs. For the purposes of this debate, let's stipulate that each of the things we're talking about were separately created. Let's just start there. According to the definition of dinosaur, birds are dinosaurs. Dr. Dino agree or disagree, right? And not only did he obviously disagreed, he was unable to provide a working definition for
Sure.
dinosaur. For like, if I showed you a skeleton, how would you tell me it's a
Of
dinosaur, right?
course.
He couldn't
Yes.
do that. And it was
Right.
just 45 minutes of Dapper Dinosaur hammering Kent on giving himself the nickname Dr. Dino, yet not knowing the definition of dinosaur. It
That's
was
beautiful.
wonderful.
Where was that?
I don't know where that was, but if you
Okay.
search, it's out there.
It's out there. All right.
You
I'm
can
gonna have
find
to
it.
look that up. All right.
Yeah, it's
I'm
around.
gonna I
It's
will
one of the classics.
that sounds great. I will definitely link to that below just so that other people coming to this video will be able to find it. The other thing I'm gonna link to is the 2009 paper that you mentioned as well. I think that would be helpful to put on there. And yeah, do you have anything else that you'd like to wrap up with, or we've been going for an hour and 15 minutes, and I super appreciate your feedback here. I've got some great ideas and about what I'm going to do. I think that I may end up doing that, just conceding the entire thing. That's kind of, I was going to say,
Yeah,
like,
just
we're not arguing about it, but I might just be like, we're conceding that.
yeah.
It could be that there was an alien that came and brought some sort of proto carnivore down to Earth. I'm arguing then from that point on that it evolved over the last 65 million years ago into these different groups. You are arguing that it all happened in 6,500 years, and that 4,000 some years ago there was a flood, and that all of the descendants of these different kinds diversified within a few hundred years after that event. Explain yourself.
Bingo.
Yep.
That's how I would do it. Yeah,
Yep.
I think that's. By the way, I'm putting in our private chat here. I just put the. Oh, wait a minute. That was just the words. That wasn't the actual. Hang on.
Oh.
Copy link. Here you go. I'm putting the link for the debate I just mentioned in the private chat there, so you have the link.
Good.
It's a classic, absolute classic.
Awesome. Thank you.
Yeah, I don't have anything to add. I think this is great. I think this paper,
awesome.
With the just this huge data set of mutation rates and focusing on a hyper specific group is a great approach, because the problem with Kent is not that he knows what he's talking about. The problem with Kent is that he says the same freaking thing no matter what the topic is. Like,
yes.
You could just play Kent's opening and play a game of like, guess what the debate topic was, and you'll lose every time. You have no idea,
because it's the same thing. Yeah,
it's the same thing.
And when I wrote you to say like, oh boy, I don't necessarily want to spill the beans too much. It's not like he's ever going to see this, but even if he watched this, he wouldn't change anything, and he wouldn't even understand most of what we're talking about. So
so the point is, make it clear enough for your audience. Like, here's the paper, here are the rates, and that simple equation. Just like, we're not doing complicated math. Just, you got to fit this much stuff into a box this size, and you can't do it. And Kent can't explain that, and then just hammer them.
Right.
And concede the right. I would start off with like, for this debate, all this stuff was created. I don't care, but we're going to start with animals and then go.
That's right. Yeah. Yes, exactly. Yeah. And I think that's the reason why I picked the topic. That's just that we can cut out basically 90% of all of his material.
Yeah,
and I'm very curious to see what he does, but it's going to be a shit show, I'm sure.
It always is with him, and especially if you knock him off script, he gets really flustered and angry, and
he's going to want to like, oh, I got to get up early tomorrow.
Yep.
Yeah. All right. Well, good. Thank you so much for doing this. I really appreciate it. And once again, obviously, I've already said it twice. I'm going to say it a third time. Go and subscribe to Dr. Dan. It's really great material, and it's super informative. I always learn something, and so I super appreciate it. Thanks for everyone for watching this, especially the live chatters. A lot of friends here that are watching, and I appreciate that. If you can, of course, want to subscribe, that's great. And I will check you later. Bye, everybody.
Thanks, everybody.